engine of evolution. Better known as natural selection, came from the concept of Darwinism. Prior to Darwinism, thinking in regards to the emergence of life largely rested on the idea of intelligent design. This is the idea that life is perfect in the image of a creator. Evolutionary biologist Charles Darwin went on to challenge the concept of intelligent design by proposing the theory of natural selection. Early problems with natural selection highlighted flaws in nature due to limited access to advanced research and scientific methods. Natural selection is commonly misunderstood by the general populace. For example, when people think of natural selection, they think of survival of the fittest, which is really a debunked theory known as Lamarckian evolution, which tends to oversimplify the concept of natural selection. Natural selection instead is a mechanical process. A process refers to genes' ability to thrive in an ideally suited environment, which allows them to become more frequent in a population through successive generations. This fitting of variation by natural selection generates adaptations. Natural selection also works on the population level. Donald Kaufman, professor at Kansas State University, showed that mice survive better when their fur matches the color of the soil in which they live. Kaufman built large outdoor enclosures, some with light soil, others with dark soil. In each cage, he put an equal number of mice with dark and light color coats. He then released a very hungry owl into each cage, returning later to see which mice survived. As expected, mice with coats contrasted conspicuously with the soil were picked off more readily, showing that camouflaged mice really do survive better. This experiment also explains a general correlation that we see in nature. Darker soils harbor darker mice. Their genetic composition went on to reproduce in the next population, had an increased proportion of genetic variance and enhanced survival and reproduction. Adaptation in a nutshell. Adaptations have three main components. The starting population must be variable, or else it will not evolve. The population must also be a proportion of variation, or else it will not properly evolve. This is usually done in the form of mutations, which can be good in the fact that they can enhance the population's fitness, or bad and decrease the population's fitness, but most commonly, mutations are neutral and have no effect on the fitness. The final component is genetic variation. Genetic variation on a species must be able to leave offspring for adaptation to fully occur. Evolution by selection is done through the process of randomness and lawfulness. Randomness is the occurrence of a mutation that is able to generate an array of genetic variation which can be good or bad. Lawfulness contrasts by ordering variation, keeping the good while slowly removing the bad over time. Adaptive traits. Adaptive traits are a step-by-step scenario for evolution of a trait that helps raise the fitness and increase the reproductive output of the possessor. Look at the traits, and it also looks at the traits of species that are useful from a member from a second species. Adaptations always increase the fitness of an individual. Adaptations do not always increase the fitness of a population. For example, in lions, if infanticide is a common practice where males will kill from an outside group, will kill the offspring of another male so he can mate with females and increase his fitness. This hurts the overall population of lions.
Evolution without selection. Most biologists define evolution as a change in the proportion of alleles in a population. Every individual has 2 copies of each gene, which can be identical or different. For example, if you have an AB blood type and produce only one child, there's only a 50% chance it will get your A allele, and a 50% chance it gets the B allele. In small populations, the proportion of different alleles can change over time entirely by chance. Eventually, the resulting random walk can even cause genes to become fixed in the population, meaning it reaches 100% frequency, or alternatively, get completely lost. Genetic drift is the random change in frequency of genes over time. Drift is a random process, while selection is the antithesis of randomness. Genetic drift can change the frequencies of alleles regardless of how useful they are to their carrier. Selection, on the other hand, always gets rid of harmful alleles and raises the frequencies of beneficial ones. Genetic drift can't cause the evolution of adaptations, meaning it could never build a wing or an eye. Genetic drift can cause the evolution of features that are neither useful nor harmful to the organism. Genetic drift is powerless to create adaptations, but can overpower natural selection. Especially in small populations, the sampling effect can be so large that it raises the frequency of harmful genes, even though selection is working in the opposite direction. Variation in DNA or protein sequence may be, as Darwin said, neither useful nor injurious. Such variants are liable to evolve by drift. For example, some mutations in a gene don't affect the sequence of the protein that it produces, and so don't change the fitness of its carrier. It is possible that many externally visible features of organisms could evolve via drift, especially if they don't affect reproduction. Animal and plant breeding. Animal and plant breeding is a good parallel to natural selection. Take the domestic dog, a single species that comes in all shapes, sizes, colors, and temperaments. Dog breeding validates 2 of 3 requirements for evolution by selection. 1. There was ample variation in color, size, shape, and behavior in the ancestral lineage of dogs. 2. Some of that variation was produced by genetic mutations that could be inherited. Evolution in the test tube. In a laboratory, we can naturally observe evolution by exposing a captive population to new environmental challenges. Microbes like bacteria, which can divide as often as once every 20 minutes, allowing us to observe evolutionary change over thousands of generations in real time. These tests demonstrate all 3 requirements of evolution via selection. 1. Variation. 2. Heritability. 3. Differential survival and reproduction of variants. Barry Hall and his colleagues at the University of Rochester began a study by deleting a gene from E. coli. This gene produces an enzyme that allows bacteria to break down the sugar lactose. The geneless bacteria were then put in an environment containing lactose as the only food source. Initially, they lacked the enzyme and couldn't grow. But only after a short time, the function of the missing gene was taken over by another enzyme that, while previously unable to break down lactose, could now do so weakly because of the new mutation. Eventually, another adaptive mutation occurred that increased the amount of new enzymes so that even more lactose could be used. Finally, a third mutation at a different gene allowed the bacteria to take up lactose from the environment more easily. This experiment showed evolution of a complex biochemical pathway. The experiment also showed that natural selection can promote the evolution of complex interconnected biochemical systems, and also selection does not create new traits out of thin air, meaning it produces new adaptations by modifying pre-existing features. Resistance to drugs and poisons. Drug resistance, not often realized, is the best example we have of natural selection in action. In the 1940s, penicillin was a miracle drug, especially effective at curing staph infections. Now, 70 years later, more than 95% of staph strains are resistant to penicillin. What happened was that mutations occurred in individual bacteria that gave them the ability to destroy the drug, and of course these mutations spread worldwide. Other species have adapted to human-caused changes in their environment. Insects have become resistant to DDT and other pesticides. Plants have adapted to herbicides. Fungi, worms, and algae have evolved resistance to heavy metals that have polluted their environment. When a population encounters stress, such as change in salinity, temperature, or rainfall, natural selection will produce an adaptive response.
Selection in the wild is a slow process, and as a result is almost impossible to observe in real time. The best chance of observing natural selection in action is to look at plants and animals with short generation times. These plants and animals should be adapting to a new environment, whether via invasion or in response to severe environmental change. One potential difficulty in the observation of natural selection is that a large part of the process doesn't actually cause any change. In situations where a species has evolved traits that are suitable for its environment, selection will only cull members of the population that fall outside the optimum range for that trait. Even so, there are real-time examples of the process. For instance, in 1977, a severe drought reduced the supply of seeds on Daphne Major, an island in the Galapagos. The medium ground finch, which lived on the island, preferred small, soft seeds. However, the drought limited this supply, and so in order to survive, the finch was forced to eat the larger seeds. Larger beaked birds were more fit to eat these seeds, and so left more offspring. In only one generation, the finch's beak size increased 10%. Most people know that selection can easily influence the size of a given trait. But what about truly complex adaptations? To start, we know of no other natural process that can explain the creation of a complex adaptation, nor do we know of any adaptations whose origins cannot be explained by natural selection. The blood clotting pathway in vertebrates is a good example. A wound is opened, and a given protein in the blood sticks to another protein, starting a cascade of reactions with at least 16 steps. The end result is a clot consisting of some 20 proteins. We know from genetics that gene duplications occur during replication, and that duplicated genes evolve along separate pathways. Furthermore, a key protein in the clotting process is fibrinogen, which is present in the blood. The last step of clotting blood involves cutting up the protein into shorter protein strands called fibrins, which form the main component of the clot. Fibrinogen occurs in all vertebrates as a clotting protein, making it likely that there was an ancestral protein from which the fibrinogen evolved. Some raise the question of whether there's been enough time for these adaptations to occur, but rates of change in animal size and shape are much faster than rates of fossil change. In some cases, they're 500 faster, as in during colonization. In the lab, they're oftentimes 1,000,000 times faster during selection experiments. Average rates of evolution in colonization studies indicate that in 10,000 years a mouse could evolve into the size of an elephant. Proponents of intelligent design often point to the vertebrate eye as evidence of their viewpoint. Given its complex arrangement, all of its constituent parts must work in concert for the organ to function, but the complexity of the eye can be broken down into a series of small adaptive steps. A possible sequence of changes may be, first, that the simple eye spot was made of light-sensitive pigment. The skin then folded to form a cup that protected the eye spot and allowed it to better localize the light source. There was a further narrowing of the cup's opening, then a capping with a transparent lens. Eventually there was an evolution of a retina, an optic nerve, and so on. It's estimated that the entire process would have taken less than 400,000 years. Eyes have evolved independently in at least 40 animal groups. And that concludes our presentation on the engine of evolution.